We should go ahead and get started. I'm Darren Wallace from the University of Kansas Medical Center. I'm going to be kind of running the meeting here. I want to welcome everybody to the first scientific roundtable since the expansion of the PKD RRC. So now, in addition, sorry, Carlene, can you please mute? So in addition to the founding members of the RRC, University of Maryland, University of Alabama, Birmingham, the University of Kansas, we've now expanded to include the Mayo Clinic, Cornell University, and the University of Washington. So as the RRC will continue to provide the reagents and resources that we generated in the first 5 years, but we also have a new resource development core that's chaired by Owen Woodward at University of Maryland. And the RRC would like to have input from the research community on the type of resources and reagents that would be most useful to contribute to the PKD field. So before our roundtable discussion, Dr. Woodward would present a little bit of information about the resource development core, and then we'll proceed with the presentation. So, Owen.
Thanks, Darren. So, I mean, one of the changes that have happened with the PKD RRC 2.0, I guess we could call it, is that we now have a kind of a centralized resource and development core. Instead of each university kind of running their own show, we've kind of centralized that with a primary goal is to facilitate technology specifically that comes from requests and needs from the research community. So everybody on this call, essentially, as opposed to just kind of top-down pronouncement of what we think is important. So we really have to hear from you about what the community needs for progression and our understanding of PKD in order for us to facilitate the development of these new resources. So over the next few months, you will be kind of, I wouldn't say bombarded, but you will be kind of given some opportunities to give us input back under a couple of different guises. So, one obvious way is that anytime you guys have an idea, and this doesn't have to be like a fully formed project proposal or anything, it could simply just be a 1 or 2 sentence, I think we kind of need something like this. You could just send it directly to any of the members of our resource and development working group, which I've kind of listed in the bottom right-hand side of this slide. So you can always do that. Another way that you can provide some kind of input and feedback is that in the next couple of months, we're going to be sending out an email survey. I know we all get lots of emails, but this one hopefully will be fairly concise. So we'll just be asking for, again, like a 1 or 2 sentence, suggestion about, your greatest needs for your research, for instance. We also will be reaching out and creating focus groups of 8 to 10 people where a little bit more in kind of in-depth questioning of the community of what they really need and what that would look like. The PKD RRC website will also have a tool built into it where you can just simply input an idea. There'll be just, a place for you to type information. And again, this could be, from fully formed, super nice proposal for a project to simply just 2 sentences, we needed this new cell line, blah, blah, blah. And then finally, for those who do really have these like complete project ideas for a new research tool, one of the possibilities is that through the research and development pilot grant program, you might be able to get that funded. And of course, we would help facilitate getting all the resources that the successful projects would need to get that to work. Now, of course, at the end of the day, not everybody's ideas will be developed, but our goal is to kind of find consensus among the community about what is really necessary. And of course, with our kind of limit of resources, we will decide which ones to develop, and then as soon as they're available, we'll distribute them back to the community for use. So anyway, so just be looking out for all these ways that we're going to contact you in the near future, and please give some input. Again, this could be very specific to what your lab needs to progress its work, because if you need it, then I suspect lots of others will need similar types of reagents. All right, thank you very much.
Thank you, Owen. That's great. The only other announcement that we have before we get started is that please be aware that there's a new RFA. I think it just actually went out. I saw an email that hit my inbox. So you might be looking for the RFA uh, for um, for the pilot and feasibility grant. So I'd like to introduce our speaker today, Dr. Vishal Patel. I think most of us know him quite well. He's a professor of medicine at UT Southwestern. He's also the director of the Comprehensive Polycystic and Genetic Kidney Disease Clinic and Research Center. Dr. Patel received his MD degree at medical school in Gujarat, India. He came to the United States to do a residency at Northwestern University Medical School. And then in 2010, he moved to Dallas and did a nephrology fellow with Peter Igarashi at UT Southwestern, subsequently establishing his own research program focused on non-coding RNAs, particularly microRNAs and their role in regulating the message levels for polycystin 1 and polycystin 2, the genes involved in PKD. Dr. Patel is really a world-class physician scientist. His work on miR-17 led to partnerships with Regulus Therapeutics and the development of farabursen and the discovery to human testing. And his talk today is farabursen ups and downs and translating microRNA biology in PKD. So take it away, Vishal.
Thank you very much, Darren, for the kind introduction, and Terry for inviting me to speak. I'm honored to kick off this seminar series for the newly appointed and some reappointed U54 PKD centers. But I want to start by actually congratulating you, Darren, Terry, and all the other PIs associated with this U54 consortium on this new, fresh 5-year award cycle. Just personally, I and the part of the broader PKD community I am genuinely excited to collaborate with the course over the next 5 years, and to move PKD research forward in ways that truly benefit patients. And then finally, I'd remiss if I didn't thank the NIH and the NIDDK for their continued support for this initiative and for PKD research more broadly. We are deeply grateful for that. So today, what I thought I'd do is, since farabursen, an anti-miR-17 oligonucleotide, is poised to start a pivotal registrational phase 3 trial in 2026, I thought I'd give you the backstory behind this compound and our trials and tribulations of the journey that took us from preclinical discovery to clinical research. So first and foremost, though, my disclosures as they relate to farabursen. One, we have a long-standing collaboration with Regulus Therapeutics, and now more recently Novartis, with whom we've co-developed this drug. We hold 2 patents related to this drug that have been licensed to Regulus Therapeutics and Novartis, and we receive licensing fees and royalties through those patents. Of course, I'd be discussing farabursen throughout my talk. All right, so the real reason why we're here is because of this devastating disease, literally and figuratively a big problem, affecting millions of people worldwide and representing the most common monogenic condition of monogenic cause for kidney failure in the United States. And so the overarching goal for us, as it is for many of you, is to build on the success of tolvaptan and ask whether, rather than simply slowing the rate of progression, can we actually arrest disease progression in this condition? If you're feeling more bullish and ambitious, perhaps pushing the envelope even further, we could ask, can we revert disease pathology in this condition? So we guided by these large, overarching, pie-in-the-sky questions, embarked on a drug development program. And I may be a bit biased, and this may sound like I'm preaching a choir, since most of you guys are biologists. But the starting point for any drug development journey is not proprietary chemistry or some inaccessible drug development platform. It is good old-fashioned foundational biology. And we chose what we think of as David and Goliath biological axis of microRNA-mediated mRNA repression. MicroRNAs are only a fraction of the size of their mRNA targets. Yet, if the base pairing between the microRNA and the mRNA is correct, this leads to these tiny molecules slay the giant mRNAs. So when we began this work in 2009, this represented a relatively new mode of post-transcriptional gene regulation. And perhaps just as importantly for me as a new investigator at the time, it offered an opportunity to work in fresh and understudied area. That said, elegant biology does not automatically mean a good drug candidate. From the outset, though, we viewed microRNAs as pragmatic therapeutic entry points for the following reasons. First, it was becoming clear that most microRNAs are dispensable for baseline homeostasis, and instead act in a highly context-specific manner as fine-tuners of gene expression. This suggested that there is some degree of built-in safety in targeting these molecules with chemical approaches. Second, individual microRNAs simultaneously regulate multiple correlated pathways. So in the right biological context, this provides a meaningful leverage. You can modulate large disease-relevant networks all at once rather than going after a single node. Subtly, but all at once. Finally, because microRNA activity is dictated by the nucleotide sequence, the seed sequence, drug discovery does not rely on massive empiric small molecule screens. Instead, it can be approached rationally using relatively small libraries, as you'll see towards the end of my talk. Relatively small libraries of complementary oligonucleotides. So for all of these reasons, we decided to chase microRNAs and see if they could be viable drug candidates for ADPKD. So the first order of business was deciding which microRNA to go after. And we started with a microRNA microarray screen. So yep, that's not a verbal typo. In the era of single-cell genomics, I'm ashamed, but slightly proud also to say that microarrays, this screen has been the best I've done in my career. We integrated PKD1, PKD2 knockout kidneys with human ADPKD samples graciously provided at the time by Darren Wallace. I think this predated the RRC, but I still give him and the RRC credit for it. We've identified a small set of microRNAs that were consistently deregulated across both species. One stood out immediately, and that was the microRNA 17 family of microRNAs. The reason why microRNA 17 was intriguing to us at that point was because miR-17 was already known to drive by then pro-proliferative pathways in cancer, many of the same pathways that are already active in ADPKD. And in fact, it had earned a nickname, OncoMIR1. So our thinking was simplistic. If miR-17 is bad in cancer, it is highly unlikely to be doing anything good in ADPKD. And so just let's chase it. So next we asked simple question, prove sufficiency, necessity of this microRNA in the disease context that we're interested in. And so here I'm showing you some necessity experiments. We use these commonly used kidney-specific PKD1 knockout mice PKD1 and PKD2 knockout mice, and found that removing miR-17 in this setting improved creatinine, reduced proliferation markedly, in fact. And this very aggressive mouse model, this is KSP3 PKD1 flox mouse, it improved survival. We saw a similar benefit with PKD2 knockouts, where again, it improved creatinine, reduced proliferation, and extended survival. So taken together with other supportive mechanistic work and consistent results across additional orthologous and non-orthologous models. In fact, we deleted this microRNA in a total of five ADPKD models. We came to this simple mechanistic framework. PKD1, PKD2 mutations lead to activation of c-Myc, which in turn induces miR-17. miR-17 then reinforces the pro-proliferative dysregulated gene network in ADPKD through targeting critical mRNAs. Importantly, deleting miR-17 does not target a single pathway. It reduces proliferation by simultaneously damping multiple disease-relevant pathways. Exactly the kind of leverage you need in a disease like ADPKD with pleiotropic downstream signaling consequences. So this is nice, straightforward, simple.
Sorry, Vishal, you're taking questions now or at the end?
Later.
Later?
Yeah.
Thank you.
I hope you don't mind. If you have a quick, brief question, I'm happy to entertain.
Very quick, because every time we send a grant talking about cell proliferation, we get reviewers saying cell proliferation is not important in PKD. Since you put the slide right there at the beginning of the talk, I just wanted if you can address that. If not now, while you're talking, and what do you think about that?
I'm not that reviewer for that much I can guarantee. I mean, it is well established that the cells do grow. The cells outline the cysts do grow, which one of the reasons why the cysts grow. There are other reasons, of course, but proliferation is important. Okay, so while this was a straightforward, simplistic view of how miR-17 operated in the context of polycystic kidney disease, it actually ignored the elephant in the room, because from very beginning we knew that one of the key targets of miR-17, certainly as it relates to ADPKD, which is the disease that we're interested in, were PKD1 and PKD2 itself. So shown here is the PKD1 3' untranslated region, PKD1 mRNA 3' untranslated region, and the red box is showing you cross-species conservation and the prototypical miR-17 binding site embedded in the 3' UTR. This, of course, was a bioinformatic prediction at the time, but we've gone on to validate it multiple different ways, experimentally in both cellular systems and in vivo models. And indeed, miR-17 comes and binds to this exact nucleotide sequence, and there's a reciprocal relationship with miR-17 going up PKD1 and PKD2, which I'm not showing here, going down. So what this does is it added an additional mechanistic layer to how miR-17 may be exerting its pathogenic effects in ADPKD. And with regards to its interaction with PKD1, or for that matter, PKD2, this is how we thought of it. In ADPKD, one copy of the PKD1 gene is inactivated, and that's inherited from the affected parent. And that's shown here as a gray lightning bolt copy. Where present, the wild type intact copy continues to make mRNAs, but those transcripts get bound by miR-17 and are repressed by this microRNA. In this scenario, you can think of miR-17 essentially functioning as a somatic epitranscriptomic second hit, reducing the gene dosage and working on top of the genetic inactivation. Importantly, the reverse scenario would be that by deleting or therapeutically inhibiting miR-17, we could potentially unmask and harness the remaining functional allele where present and potentially compensate for the copy that was lost. So this is the intriguing idea of the miR-17 PKD1, PKD2 dosage regulation. But how do we test it? Remember, I just mentioned that the models that we had used thus far involved biallelic inactivation of PKD1 or PKD2. So there was no residual PKD1 around to test this idea. So here we leveraged the RC mouse model that was developed by Katrina Hopp and Peter Harris at the Mayo Clinic. And we kind of improvised on this. In this model, the RC mutation is present in the germline and therefore all cells of the body, while the second PKD1 copy carries LOXP sites flanking critical exons. Kidney-specific Cre deletion converts this mouse into tubular epithelial-specific monoallelic RC- state. So the tubule cells are monoallelic now. As a result, and as expected, this leads to reduced PKD1 gene dosage, activation of miR-17, and a reliably penetrant and aggressive ADPKD-like phenotype. This is a P18 mouse. In this setting, when we remove miR-17, we see profound effects with improved kidney architecture, fewer cysts, and normalish looking kidney size. But perhaps more gratifying was that we also observed increased PKD1 gene dosage. And so from a genetic makeup, considering the genetic makeup of this mouse, for all practical purposes, the remaining copy, the RC copy, was producing this extra PKD1. And that indeed was having a beneficial therapeutic output as shown by improved cystic phenotype. But moreover, this inhibiting miR-17 de-repressing PKD1 appeared to have a disease arresting effect, as shown here by the survival analysis, where removing miR-17 essentially normalized lifespan in this mouse model. Please note that we followed these mice for up to 2 years, and so which kind of is close to the normal lifespan for this model. So this was very exciting, of course, for us, and perhaps foreshadowed the transformative potential of restoring PC1 or PC2 in the context of ADPKD. So with this now, we were turning to an updated model, now including both PKD1 and PKD2 in it. We see that miR-17 does more than just amplifying downstream procesogenic pathways. It also directly hits the root cause by reducing PKD1 and PKD2 dosage, further amplifying this pathogenic circuit. Importantly, inhibiting miR-17 offers a way, perhaps, to harness the residual PKD1, PKD2 where present, potentially restoring functional gene dosage and slowing disease progression. So with the biology, of course, I'm not showing you all the mouse work that we've done, but at this point, we felt that the biological validation was good enough and that we had a decent mechanistic framework to ask the next big question, and certainly a big step for me, not having done this before, whether we can make a drug out of this biological axis. It is at this point that we were able to partner with a biotech company, Regulus Therapeutics at the time. It was a publicly traded company with expertise in microRNA-based drug development. And we were able to launch a collaborative initiative with the goal of developing an anti-miR-17 drug. So this is the bottom line. As I mentioned earlier, the therapeutic target of microRNA involves designing entamid oligonucleotides with complementary sequences. So unlike traditional small molecule screens, this library does not need to be enormous. Because the primary variable, the sequence, that is fixed, we are bound by what the nature's already made. And so we have to produce a complementary sequence to the seed sequence of the miR-17 family. And so essentially what the library involves is working with this constant complementary sequence and modifying the backbone with various chemical modifications that will improve stability, perhaps favor kidney-targeted delivery, dampen immune activation once this foreign RNA is introduced into the body. And surprisingly, this screen was of only 199 oligonucleotides. And out of that, this one rose to the top. It was called RGLS 4326. It is a 9-mer with complementary sequence to the miR-17 family seed sequence. The red, blue, and the green show the chemical modifications that were made at the backbone to impart PK properties, drug-like PK properties. And with the compound in hand, 2 questions, 80 PKD specific questions needed to be addressed. First is there effective kidney delivery? Oligos until this point were being used primarily for liver disorders, and a kidney oligo, as far as I know, was not being developed at the time. So this was still delivery to kidney was still a barrier back then. And then second, of course, because this is a nascent drug delivery program, we have to think about the downstream phase 1 that you'd want to do. And so defining pharmacodynamic readouts that both capture the miR-17 or anti-miR-17 activity, but hopefully also double as 80 PKD-centric disease progressing biomarker. So we had to search for that, of course. That was not as difficult, frankly, based on all the mechanistic work that we had done. We thought that PKD1, PKD2 would make our obvious top choices for looking for both miR-17 activity, but also 80 PKD progression. So first, the delivery data. Here, what I'm showing you is an autoradiograph from a mouse that was administered subcutaneously, a radio-labeled RGLS 4326 compound. The red signal denotes radioactivity, and wherever it's bright red is where the drug went. And you can see the glowing thing inside the body is the kidney, and that's shown here graphically. In fact, the drug accumulated in the kidney at approximately 8-fold higher level than the next highest tissue, which is the liver. And so this distribution is completely flipped compared to what the other oligos were being tested at the time, where liver is first, followed by other organs, including the kidney. We believe that this unique biodistribution, which is atypical for the oligonucleotide drug class, reflects a combination of the backbone modification and, in large part, the very small size of the oligo. Remember, it's only a 9-mer. The other oligos that were being developed and were marching towards FDA approval were 18-mers, 20-mers, and even higher. So this 9-mer, even though it's oligonucleotide, perhaps is a separate subcategory of oligos based on the size. Next, on to PD. Once in the kidney, the drug gratifyingly restored PKD1 and PKD2 mRNA levels in the RC minus mouse, the monolytic ADPKD mouse model. These studies were done after cyst had already formed, and so the drug actually penetrates the cysts and appears to have biological activity. The plots show PC1, PC2, full-length PC1, PC2 depression in primary human ADPKD cell lines, showing cross-species depression of this centimere. Again, I have to acknowledge Darren for graciously providing these samples. This is one of many that he has sent us over the years, and I'm thankful for his collaboration. All right, so delivery check, PD good, and so efficacy, of course. In terms of efficacy in collaboration with Regulus Therapeutics, we have actually conducted more than 30 independent preclinical studies. Of course, I'm not going to show you all of those. Today, what I'll do is highlight one that I find particularly exciting. It's one of the newer ones, and it's unpublished. In this experiment, we used an inducible reduced PKD1 gene dosage model. One allele carries the RC mutation, while the second allele is deleted postnatally using the doxycycline inducible KSPRTT hetero system. Why do this? This was done because this design allows us to establish pre-treatment baseline using non-invasive approaches such as MRI, shown here. These kidneys are about 8-fold enlarged compared to an age-matched normal non-cystic mouse. Creatinine is already elevated, and so for all intents and purposes, you could say that this is a class D, perhaps a class E ADPKD in mice. In mice treated with PBS, the disease progresses as expected, the kidneys grow, and at later stages, there's fibrotic remodeling with slight shrinking of the total kidney volume. In sharp contrast, treatment with Argelis 4326 leads to an initial arrest of disease progression followed by active cyst resorption, with volumes falling an impressive 40 to 50% below the treatment pre-treatment baseline. So this, truly shows pharmaceutical reversal. Now, I admit that this mouse model is tailor-made for this drug to work because it's monoallelic. Nevertheless, it does provide proof of principle that disease reversal under the right circumstances is possible. Too many images. Here are the money shots pre and post. Oops, sorry. Never mind. And the one extra point I'd like to make up around here is that this work, I feel, nicely complements Steve Somlo's genetic reconstitution work, and obviously underscores the transformative potential of pharmacologically restoring PKD1 and PKD2 dosage, either through residual depression or providing exogenous PKD1, PKD2, and many of the other ways that people are thinking about. So with this preclinical work, on to humans. And the question is how do we test this idea? Remember, we are doing a phase 1, which is limited in scope and duration and all of that. And ours was a nascent drug development program, and there was little blueprint, frankly, for designing phase 1 studies for this for ADPKD. But based on our preclinical data, we reasoned that the most informative axis to interrogate was perhaps the miR-17 PKD1 PKD2 axis. And so mechanistically, mutations in PKD1 PKD2 increase MYC and miR-17, which represses its downstream targets, including PKD1 and PKD2, resulting in reduced PC1, PC2 protein levels. Prior work from Chris Ward, Murray Hogan, and others have shown that PC1 and PC2 are abundantly present in urinary exosomes, So we therefore viewed urinary exosomes as practical, if somewhat crude, window into looking at what PC1, PC2 protein levels were in the kidney. Hypothesis is straightforward. In patients treated with RGLS, disrupting the miR-17 axis would relieve the repression, raise PKD1 and PKD2 protein levels, and this would be reflected via increase in detectable PC1, PC2 in the urinary exosomes. Okay. So with that said, the trial was designed to test this. This involved inventing the wheel a little bit of getting the PC1, PC2 exosomal assay up and running through a CRO, which I give full credit to the work from Chris, but then improvised by the Regulus team. Excitingly, we saw data that was exactly what we were hoping for. Compared to pretreatment baseline, our RGLS4326 treatment increased PC1, and I'm not showing you PC2, levels by nearly 60% in individuals with class C, D, or E ADPKD. And so for us, this represented an important proof of principle that pharmacological inhibition of miR-17 could de-repress PKD1 and PKD2 in humans, and can be measured through urinary dynamic changes in the urinary exosome. That said, there are clear limitations with this trial design. This is a small study, N of 9 only. We tested only a single dose, 1 milligram per kg, with only 4 injections, so small duration, and of course, no placebo arm. We didn't think of these as oversights, perhaps deliberate constraints, because we were simply looking for initial signal-seeking study. And the logical next step was going to be a longer duration, multiple ascending dose, randomized, placebo-controlled trial. As is routine in preparation for these larger studies, and for regulatory filing, Regulus Therapeutics initiated a chronic dosing preclinical safety program. This is where we hit a roadblock. In a subset of animals, we began to observe classical signs of CNS tox. Follow-up studies revealed that this tox emerged in large part due to very high cumulative doses on the order of around 2,000 milligrams of total drug exposure. You can see various different dosing regimens that all essentially amount to about 2,000 and produce CNS tox in a small subset of animals. Okay, at this point, I have to mention that 2,000 mg cumulative dose is about 50 to 100-fold higher than the therapeutic dose that we had been using in mice. So the safety margins were still there, but there is dose-limiting tox. And this was obviously promptly reported to the FDA, who then promptly put in a partial clinical hold on the clinical program, which means that the company was allowed to continue trials, but not exceed 1 mg per kg dosing, and that was going to be their ceiling. Of course, that's a terrible handicap, especially when we didn't know what the effective concentration was going to be in humans. And so this is often the point where programs get shelved, deprioritized, abandoned, essentially throwing the baby out with the bathwater. But instead of walking away, I actually want to give real credit to the team at Regulus, who believed strongly in the underlying biology and decided to dig deeper. So the key question they asked was, yes, this is toxic. Why is it toxic? And this work is now published about 2 months or about a month and a half ago, came out in Nature Communications, so you can read our sob story there. But the The bottom line and the snapshot of the talks is shown right here. The answer came from electrophysiology, and here what I'm showing is structural biology. This is cryo-EM showing the EMPA receptor, which is critical for fast excitatory synaptic transmission in the brain. Curiously, the blue is RGLS 4326. It's sitting inside the EMPA receptor complex. Specifically, you blow it up, you see a single, the single G at the 3' end. You may have missed it because I moved too fast on my initial oligonucleotide RGLS design, but there's a G at the 3' end. And that was directly engaging with the ligand binding domain of this receptor and competing with the natural agonist glutamate and promoting channel closure. Turns out G contains a hydrogen bond acceptor at the 6th position. And this was allowing to pair with critical residues in the ligand binding domain and providing the structural explanation for both the MPR inhibition and CNS tox. And so shown in a cartoonish way, RGLS 4326 has an off-target aptamer-like effect, not to an RNA, to a protein, which still blows my mind. And at a concentration of about 2,000 cumulative dose, just enough of it trickles through the blood-brain barrier to mess with this critical receptor. So if G is the problem, there could be a simple solution. Take it out, switch it. Put a different nucleotide that does not have a hydrogen bond acceptor in its structure. And that is indeed what they did. They switched the G to an A. And that produced the next generation compound, at the time called RGLS 8429, and now called farabursen. Everything else remains exactly the same. This one nucleotide change, but the backbone chemistry remains unperturbed. In fact, the changes in backbone chemistry were tested in the screen with the MPR receptor interaction and were found not to be involved in that. And so it was squarely pointing towards the G. So remarkably, this simple substitution took the tox away. With 8429, no dose-limiting tox was observed, even at doses that were 7-fold higher than the toxic dose for 4326, which itself was about 50 to 10-fold higher the therapeutic dose. And so with this new molecule, farabursen, retained all the properties we cared about: kidney-enriched delivery, derepression of both PKD1 and PKD2 in mice and in human cell lines. And I'm not going to show you this, but robust therapeutic efficacy in preclinical models. And so with these tox now engineered out, and after a 4-year detour, we were finally back to test this compound in patients, this time without any FDA restrictions. And so this actually gave us a true makeover and really design a decent trial. And so this ended up being a multiple ascending dose, placebo-controlled randomized trial with an additional layer, additional fixed dose open label arm, urinary PC1, PC2 levels, as well as kidney MRI were obtained at the baseline and at the end of the study. And so here are the latest data, which were presented by LNU as a late-breaking abstract at the ESN. So apologies for those who have already seen this. Compared to placebo, we observed a decent dose response in both increases in both urinary PC1 and PC2 in people that were treated with farabursen. These were 7 injections spread over 3 months, and the collections of PC1, PC2 are pre-treatment, and at the end of the study, a mean of a few collections at the end of the study. We included MRI in this as truly an exploratory analysis. For an exploratory analysis, of course, the study was not powered to detect structural changes. And considering that this was a small duration study, of course, we didn't anticipate much with the MRIs. However, we observed encouraging signs there as well. Kind of mirror image of what the PC1, PC2 levels were in the previous graphs. At higher doses in a post-hoc analysis, higher doses that including 2 mg, 3 mg, and 3 milligram fixed dose led to a cumulative increase in kidney volume compared to baseline of negative 0.1%, essentially arrested, numerically suggesting arrested kidney growth compared to placebo, which grew at about 2.7%. I want to mention these are small n's. These is not stat-sig signal. Nevertheless, it's encouraging to observe. As a result of this, a large, before that, jumping the gun ahead a little bit, but I want to revisit the model where PKD1, PKD2, the downstream pathways are in it. Farabursen is the genetic counterpart, pharmaceutical inhibitor of miR-17, which gets to the kidney, turns off miR-17 activity, and increases PC1, PC2, and impacts the downstream pathways. So as a result of all of this, a registrational phase 3 trial is planned to start in 2026. Okay, so I'm coming towards the end of my talk, and I want to reflect on our journey and some of the lessons that I've learned. I was very young and naive as I was going through all of this, stupid, some would say. Here's our journey. We started with essentially scientific curiosity, and in fact, somewhat of a non-hypothesis-driven approach. We were screening for microRNAs. We didn't have something, that we already could hang our hat on. But we were fortunate that the NIH supported our initial proof of principle work. But for a small lab like mine, you can only do so much, maybe a few genetic crosses here and there. But when it comes to making drugs, we were extremely lucky to partner with Regulus Therapeutics, which actually began in 2015. So it's now a decade old and still going. And we collaboratively set out to chemically target the miR-17 pathway. Over time, as things progressed, Regulus became the big brother with us as sidekicks essentially, with them taking on the responsibility of producing human proof of concept studies. And after all the ups and the downs that I've shared with you, they were eventually able to produce data that were compelling enough to attract an even bigger big brother, Novartis at this time, who acquired Regulus last year for around $1.7 billion. This is not just some flashy number. It's a gratifying natural progression of a drug development program, because without Novartis's acquisition, I don't think a robust phase 3 program could have been funded or executed. And so it's very gratifying to see how this has progressed. The 2 key lessons stand out. First, it takes a village. So I have been shamelessly promoting my own work and saying biologists are the next best thing since sliced bread. But it also requires chemists, in our case, oligonucleotide chemists and toxicologists, as you saw, and biophysicists, as you saw with the cryo-EM studies, and many others to bring this to fusion. And so an open collaborative spirit is critical. And in retrospect, that has been truly an integral part of our journey and collaboration with Reckless Therapeutics. And then the second lesson, which was a little frustrating for me at the time, was that while publications and academic metrics are important, and you can see we've publicly documented essentially all of our work, there are other metrics that matter as well. Regulatory, commercial, clinical milestones are equally critical. In particular, generating and protecting foundational intellectual property is essential, without which no new drug can be developed. So as academic scientists, we need to be mindful of all these components and respond to the various demands and expectations of agencies that fund our work. Maybe NIH at the start, but private investors and even public investors as you progress. With that, I'm going to conclude and acknowledge a lot of the people that have done the work, and I'll focus people from my laboratory. Jesus Alvarez, June Song, a PSTP, I showed some of their work in preclinical RGLS studies. Ronak Lakia, who is an assistant professor at UT Southwestern and directs the PKD COE at UT Southwestern, has been a longstanding collaborator. And of course, my collaborators, Edmund Lee at Regulus, who has since moved on to bigger, better things, and Tanya Valencia and my funding sources. And now I will stop, and Vasi, I'm open to all your questions.
Hi, Vishal. Thank you. That was great. We look forward to being maybe one of the sites for the trial. The thing that I always didn't understand, though, is that probably many hundreds or thousands, you probably have the exact number of genes have the miR-17 sequence. How is it not affecting lots of other things?
Well, I don't have an answer for you, but I'll hedge. So remember, these are bioinformatic predictions, okay? And it takes two to tango. So both the microRNA and the mRNA have to be physically present at the same time. So and as I mentioned, it's context dependent. And so this thing gets activated, let's say, in certain cell types, and whether its predicted targets are all also activated or present and not present, all of that is, of course, needs to be taken into consideration. That said, no, it does have a pleiotropic downstream effect. I mean, it targets PKD1, PKD2, but it targets PPAR alpha and PPAR GC1A and a bunch of other things. And so whether there will be on-target safety effects as a result of inhibiting miR-17, whether they will emerge, they haven't emerged thus far in all of our chronic preclinical testing, but whether they'll emerge in future studies, time will tell. And that is exactly one of the, that's a concern that that needs to be addressed. One way of hedging towards not having these on-target side effects would be by optimizing the oligochemistry, where it allows penetration to restricted cell types, for example, or modulating the dosing. So you go with like a lower dosing threshold, where a subset of downstream targets get de-repressed, but not all, and things like that. And those are all the things that we've kind of tested, mucked around with in the preclinical studies.
Thanks.
Hi, Vishal.
Joe?
Can I? Sorry.
Yeah, go ahead, Heng.
Okay. Vishal, nice talk. I mean, I like the approach. Anti-miR-17 would working theoretically independent of germline mutation because it actually helped the remaining wild type allele. So this is something nice. So my question is, but by the same token, would you expect in ADPKD patient cysts that the upregulation of miR-17 would target both allele, right? Both
Yeah.
allele on RNA level. So would you then expect every single cyst would have reduction of PKD1, polycystin 1 protein level? I mean, you
Okay.
can both
So
in the
yeah,
wild.
yeah. So miR-7 and the anti-miR-17 the miR-17 will target all mRNAs that have the UTR.
Yeah.
So all mRNAs that are stable enough to be engaged by miR-17, which happens in the cytosol, in the cytosol, not in the nucleus. And so you would think that the inactivating premature stop, those transcripts undergo NMD, and so those will not be accessible to miR-17 binding, and those would be the germline inherited mutations. Where present, the wild type copy will make mRNAs that will be accessible to miR-17, and that is what we seem to see in the exosomal studies because we are looking at full length PC1 and PC2, the mature full length version that gets excreted in the exosome. So, but you're right. If the mutant mRNA is stable enough and carries the UTR, it will go out and it could interact with miR-17. Yeah.
Yeah, but regardless what the mechanism, but would you not expect in each cyst you will not have, I mean, cysts, right? Because you have always upregulation of miR-17, which will going to target both alleles. So eventually you would not expect any functional polycystin 1 protein. Both allele, mutant protein, wild type protein, they're gone. Is this something you would expect?
Oh, you're saying miR-17 would repress the wild type allele, and so there will be no a protein left. Is that what you're saying? It turns it off, doesn't
Protein.
turns it down, doesn't completely turn it off. So it reduces the dosage, but doesn't completely extinguish it. So there will still be some wild type. If the wild type is present, there will still be some wild type left, even in the presence of enhanced miR-17 activity.
But it will also destroy any residual, any mutant proteins as well if they're made. What I'm saying is you're going to have no allele, no PKD1 level at all on the mechanistic level on assist genesis.
Yeah, I would say not if the wild type is present. If the one copy is gone and the wild type is present and miR-17 turns on, it will turn the remaining copy off and will not produce null, but near null is what I would say.
Okay.
Because it doesn't completely turn it off is what I'm trying to get at.
Yeah. Okay, thanks.
Yep. Lisa, you have your hand up.
Lisa, do you have a question?
Yeah, I do. So Vishal, that was a beautiful talk. And it's a tour de force that you've had tenacity to stick with this. You and I have discussed this issue before, but I wondered if you could share with the group your sort of current thinking about applying this scientific knowledge to recessive disorders, particularly because most kids with, for example, ARPKD are compound heterozygotes. And so for that group of kids that maybe has a missense mutation that doesn't work great, but it does make a protein, do you think that and there's the signature of increased MYC and there's the signature of an MR17 binding site. Do you think that this would work in recessive disorders?
Yeah, absolutely. I gave a very ADPKD centric presentation, Lisa. But if I were to look at it from a microRNA lens, I think what we are figuring out, which the broader microRNA drug development community hadn't thought of, was to not worry about the microRNA, but worry about the UTR of the gene that you really give, to sense about. And so for ARPK, it would be PKHD1. If there's a missense and if it is made, indeed, the UTR should be screened for repressive elements against matching microRNAs. If miR-17 is present there, that would be magical because we already have a drug that looks decently safe and could be tested there. But if that's not the case, you can bash the UTR to find ARPKD specific microRNA, and microRNAs. And as I mentioned, the blueprint for developing taking a pathogenic microRNA and turning into an oligo antimer is decent now, with a lot of the rules of oligo safety and all of that kind of nailed down. And so I think that would not be such a tall ask, frankly. So just quickly finish my thought. Before Regulus got acquired, they were thinking of looking at other dominant, not recessive, other dominant diseases where their copy was present, and so UMODs and TSCs, and they were looking into all of that. But pardon my ignorance, I didn't realize that there are missense PKD1 mutations and where full length could be made, and so that's something certainly that we should explore.
So, and just a sort of a corollary question, since in children, there's a whole developmental process, are the microRNAs, do they have a developmental, a temporal sequence that is developmentally specific? Because here that would be really important, both for the therapeutics and, as someone previously asked, for the off-target effects.
Yes.
That could be really detrimental.
Yes, and so this actually goes back to Josh's question. I mean, I should have answered this as well. Some of the inbuilt safety for the miR-17 targeting, but perhaps could be an issue, for example, for early pediatric conditions, is that this microRNA is indeed developmentally regulated oncogene, like classical oncogene. It's expressed highly during development, and it truly caters as the kidney reaches its mature form. And then it raises its ugly head like a prototypical oncogene at times of stress, cancer, injury, and things like that, which is how it comes about in ADPKD and multiple other cancer types. When it raises its ugly head, of course, it does not globally go up. It goes up in regions of stress and things like that. So that allows for decent way of pharmaceutically targeting it. But your point is really well taken. Without biology having completely figured, us having completely figured out the intricate biology in developmental context, whether targeting it in pediatric population is smart or not, I don't know. Yeah.
Thank you.
So Vishal, we're at the top of the hour. I just wanted to thank you for a wonderful presentation. And I wanted to let the group know that the next scientific roundtable is February 2nd. We don't yet have the speaker worked out, but we'll certainly let you know. And there were several questions in the chat. I don't know if you might have a chance to either respond to them individually, or if you'd like to stay on, we can discuss it. But before everybody left, I wanted to congratulate you on a wonderful presentation and a wonderful journey for a success story.
Thanks so much, Darren. And yes, I will stay on to answer some of the questions on the chat. Yeah.
Thank you.
Hey, Dr. Patel, this is Chen Ying from Gandela Bio. Can you hear me?
Yes.
And thank you for the wonderful talk. I just wonder, between urine and urine exosome samples, do you have any experience which sample matrix will be more sensitive or quantitative to evaluate ADPKD related biomarker changes? Say if we wanted to evaluate RNA changes or protein changes.
Well, thus far, the most validated component of the urinary ELVs are the protein, the full-length proteins. So, that doesn't mean that PKD1 mRNA and PKD2 mRNA is not in there. Is that some R&D needs to go after in that. It would be much easier to look at RNAs. Of course, protein biochemistry is quite difficult. PC1, PC2 protein chemistry especially is quite difficult. But thus far, the canonical thing that's in is the protein. I like the protein because it kind of gets goes away with this question. You did RNA, and they're like, oh, but what about the full length? And did you do the protein? So might as well just go to the might as well just go to the hard thing, which is the full length protein.
Mm-hmm. Do you think the urinary exosomes is more like have more concentrated proteins to more reliably quantify the protein changes, or comparing to urine samples?
Just plain old urine samples, I don't think is a great idea. That's dirty, of course, with a lot of urothelium and bladder and all of that stuff. ULVs, urinary exosomes, probably is probably a better way to do it. Of course, you have to define how you normalize it, and these guys have taken quite a bit of pains to figure out what the normalizer would be. I forget what that marker is. I think CD133 or something like that. But I would say ULVs rather than the entire urine.
Thank you very much.
Okay, Darren, do you want me to just read through the questions and see if people are still here and answer them? Yep.
Yeah, I can either ask you the questions.
Okay.
The next question is, are pediatric patients age 15 and older part of the phase 3 trial?
No, they're not. It's focused for adults, focused at adults. Yep.
Is there any clinical data sets that show an increase or overexpression of one or any specific miR-17 family member in the PKD patient population? Is there any clinical data set that show an increase or overexpression of one or any specific miR-17 family member in ADPKD population?
Yeah, so the data that I showed you was ADPKD samples, nephrectomy samples, of course, that were used for microRNA microarrays. I think these came from Darren Wallace. I think there were a total of about 19 individuals that were part of that analysis. Subsequently, we did immunohistochemistries and in situ hybridizations on sections, again, on ADPKD nephrectomy samples, and looked for miR-17 signal in the cis lining of ADPKD kidneys. Non-invasively, blood, urine, that doesn't seem to be a signal of increased miR-17 with disease progression or any of that. In primary ADPKD cell lines, there's certainly ample evidence to show that miR-17 is highly bioactive. And the reason why I say that is every time we throw an anti-miR-17 in it, you see an increase in PKD1 and PKD2 protein levels. So that is the level of human data that's available.
So, would you like to ask your question? There's a couple questions from Chris Chen. Would you like to just click on Chris?
Sure. Great presentation, Vishal. My question is whether or not there's any data that you could share that where Regulus or Novartis may have looked at cell lines or organoids derived from cysts known to have second hit mutations, whether or not there's any data to show efficacy in those, or whether or not within the clinical trial there's good enough imaging data pre and post treatment to suggest that you were either getting universal repression of cyst growth, or if it was like a mixture of growth and repression for different cysts.
Got it. Chris, believe it or not, every time we've looked at cell lines, primary cell lines, we've always found residual PKD1 and PKD2. So I actually haven't found a complete null cell line. You could say that I haven't looked hard enough, and that is certainly true. But we've over the years done multiple different primary ADPKD cell lines, different batches, different phases of our study. And we seem to find PKD1, PKD2 protein expression in there. So that's that. And then in terms of more qualitative info around the MR about kidney architecture with the MRIs, I know that they have the data. We and Tim Klein at Mayo Clinic has used his fancy analysis to look at some of this, but I don't think it's publicly available, not to me, but also I don't think it's been done deeply enough for public communication.
Thanks, Vishal. That's all for the chat. Wasi, do you want to ask a question?
Sure, thank you. A quick question here. So I don't know how much of the PKD1 pseudogene transcripts are there in the human compared to the mouse. And I'm assuming that MIR17 is going to be targeting also the pseudogene transcripts, right? And this is one.
Let me answer that. So the mouse does not have the pseudogenes. So that provides a very simple model to study the canonical PKD1 mRNA. In humans, the pseudogenes, as I recollect, maybe Terry and Darren, Lisa can correct me if I'm wrong, do not contain the UTRs. They stop before that. And so again, that is, I think, one of the beauties about this. It targets the UTR, which is the prime UTR, which is explicitly present in the canonical PKD1 mRNA. So yeah.
And you pretty kind of expect the same distribution in terms of uptake and biological availability of the anti-MIR17 in the human, similar to the mouse?
Yeah, I think of course, we don't have the automated radiograph that I showed you. You can inject people with radioactivity, but all of the PK analysis that have been done thus far. Remember the phase 1 trial. The primary goal of it is safety and PK and all of that. I showed you the PD and all of that, of course, but in all of the analysis they've done, it seems to follow the biodistribution seems to look like what we found in mice. In other words, around 80 to 90% of the oligo is found intact in the urines, which tells you that it once made it to the kidney, and then as opposed to the canonical larger oligos, which are not recorded in the urine at such high percentage.
Okay, sorry, one last question. I mean, I usually, when I start asking, I cannot stop, but I promise I'll stop myself after this question. So basically, you looked at the total kidney volume, which I congratulate you for doing this, because I know how much they suffered with tolvaptan when it came to TKV versus other measures. But did you also look at the cystic index? And I know Dr. Harris generated some AI tool to kind of analyze MRI images for cystic index. And do you expect to have like something similar if you stop the treatment? Is that like the cysts disappearing, or just they stay there dormant, and whenever then they kind of refill themselves with the fluid or stuff like that?
No, you're asking me to speculate, but it's okay. It's late in the hour. Most people have locked off, so I can be stupid. I would say so. About the cyst, I'll say this: the reduction in total kidney volume correlates very nicely with reduction in total cyst volume. Okay, so that is the extent of what the reduction is. What they've.
But you don't see reduction in the number of the cyst. The cystic index, not the volume.
Well, cyst index involves cyst volume, right? The MR does not have high level resolution for us to meaningfully say that the cysts went away unless you're tracking one single thing. So that's that's point number one. But you can't simply poo-poo the idea that the reduction in TKV was tightly correlated with reduction in total cyst volume, because there have been other studies where there have been reduction in TKV not because of reduction in cyst volume. And that is a bad thing. That cannot be a good thing. And so here it does at least say that you're going after the target lesion. So that's, that's sort of what I can say. In terms of what are we doing with the cyst? Are we arresting it? Are we collapsing it? I don't want to, overread the preclinical data that we have, but in the inducible model, we saw arresting initially, which would be the early small duration clinical trial. So yes, indeed, there is initial, based on the preclinical analysis, there seems to be arresting. But the preclinical data also showed that continued treatment then eventually leads to resorption of many of the cysts. In fact, most of the cysts, I would say. And so whether that will happen in people, of course, no one knows, right? That's point number one. Point number two, I have to say that the mouse model was tailor-made for this drug, a monoallelic, homogeneous monoallelic mouse, which is not the case in people. And so, I don't know, what to expect or predict. But, it all depends on how many inactivating second hits there are and all of that. So, yeah.
Best of luck.
Thank you again, Vishal. A wonderful presentation. We're going to let you go. And we'll see you again soon.
Nice seeing everybody.
Bye,
Nice seeing you,
everyone.
Lisa, Terry, Darren. Congrats again.